Naomi, you can introduce our next speaker.
Nice. Okay, yeah. So thank you everyone for the very nice discussion indeed, and for the very nice talk also, Happy. So indeed, we go and we move on to our second speaker today. Zigmunds conducts his research currently at the University of Latvia, and the long-term goal of his research group is to understand how intra-plant and inter-plant systemic signals affect and are shaped by plant beneficial microbiota in order to help plants defenses against pests and pathogen attacks. So the ambition is to make fundamental discoveries in model plants, as well as economically important crop species that could become new crop and tree models in the future. So the application of the group's research is aimed at sustainably engineering and supplementing plant microbiota to boost defenses in high-yielding cultivars, where genetic breeding or engineering may be challenging or yet limited. So Zigmunds, the floor is yours.
Thank you very much, Naomi and Raphael, for the kind invitation and introduction. I hope that you can hear me well. And today I will tell you about a very interesting story that's been the fundament of a newly emerging lab of mine, which is about underground biological internet, or more specifically, how common mycelial networks mediate interplant responses and resistance to pathogens. And this work and my group is currently based in Latvia, in the Latvian Biomedical Research and Studies Center. Although the institute focuses mostly on the biomedical research, it also has a new division on molecular ecology and biosystems, where my lab is also positioned. And one of the the strengths of the institute is that we have our in-house genomics center with sequencing facilities. We have the MGI platforms for whole genome sequencing as the DNB-seq T7, as well as the smaller platforms as the DNB-seq G400, suitable for transcriptomes or smaller genomes of small bacteria viruses. We also have the long-read platform, the Oxford Nanopore ProMethION new device, and Illumina MySeq, which is actually very good for ITS sequencing, 16S sequencing, and community characterization. And we are a new group, so we've been operating pretty much for 3 years now, since I returned back to Latvia after a career abroad, and we are the molecular plant microbe interactions group, currently based in the Biomedical Research Center, but I also have a position at University of Latvia where I do quite teaching. And that gives me access to a range of students, and you see some of them here who are also employees in my group, so they are helping me with implementing different projects. I added a QR code if you're interested to visit our group website. Please feel free to copy, follow that, and there's a lot more information. And we currently are attracting the Latvian National Academy of Science funding, as well as an EU regional development funds. And we have really strong collaborations, and here I would like to mention Professor Sunjay Lee, who is holding a position at University of Lausanne, as well as Swiss Center of Excellence for Microbiome Research, and as a newly appointed professorship at the African Genome Center. And that gives us wide connections throughout Europe. And also, if you are, after this talk, interested in any type of collaborations or even larger consortia with many partners, that's an additional network and that I can offer and build on. And Sunjay has been really instrumental at helping us to launch this work that I'm going to show you today. And actually, the story I'm about to tell you started about 400 to 450 million years ago. It's a very old story, but we only recently start to grasp what is happening there. And here you see an artist's impression of the Ordovician period, how the Earth looked like when the very first land plants crawled from water and started colonizing land. And their roots were really poorly developed, and what they think that the soil microbes, including mycorrhizal fungi, or their relatives of today, helped these plants to colonize land. And we can actually see that in the very first fossil records that have been found. And this is a fossilized, what we believe, arbuscule inside a plant structure from around that period of time in Asteroxylon plants. And since that time, these fungi have been establishing and co-evolving a relationship with plants. And what we know quite well now, and many labs have described how the one plant and fungus interactions are formed, and they help plants to absorb water and nutrients. But what is evident from this picture that you see here in this short video from a Spawn network is that they also can connect plants in one system. They effectively can colonize several plant hosts at the same time. And these interactions are really ubiquitous. If you take 1 gram of soil, you could find about 90 meters of these fungal hyphae, as you can see here in these images, with the fungal structures being on the surface as well as embedded within the plant roots. And another important feature of this system is that these networks are alive and they ship different nutrients, water, and potentially also yet unidentified small molecules and components that could help plants to exchange signals. Hence, this is the hypothesis that we've been trying to investigate in my group, is that these common mycelial networks could mediate interplant stress signals on top of their known roles in mineral nutrition. And the way we address and tackle this hypothesis is we set up experiments in such soil pots and sterile soil where we put nylon meshes, as you can see here. These are 2 pockets I will explain in a moment. And we can inoculate this soil, in fact, at the moment when we put the first seedling inside, with AMF fungus, arbuscular mycorrhizal fungus, Rhizophagus irregularis. And the system, when it's mature after 8 weeks, looks about this. So these are Medicago plants, relatives of alfalfa, a known plant model system for studying symbiosis. And so you can see that these plants are growing inside these two nylon mesh pockets. And later on, we can actually verify whether these plants have been colonized by the fungus using ink staining and really visualizing the fungal structures inside the plant root. And these meshes are actually very important. As you can see from this picture, the roots are large and the pore sizes are small. So the roots cannot directly touch each other, but the fungus can grow through these pores and actually make connections with these plants. And I will show you what we found with this system, but let me explain that we are also expanding beyond this model system of Medicago truncatula, and we are also having a joint research project with Latvian State Forestry, and we are exploring the same hypothesis, but using tree species, silver birch and hybrid aspen. In this picture, you can see these small tree plantlets growing side by side in duets in these pots, and we can also manipulate the root connections and the mycelial networks. And essentially what we do with these systems, we try to mimic biotic stress in one of the plants. We want to mimic wounding, which naturally occurs through pest attacks, such as these insect herbivores, and we can do so by forceps wounding the plant leaves. And in the created wounds, what happens naturally is that they are open space for bacterial invasions, also fungal invasions. And so we are actually using a known plant immunogenic elicitor, flagellin, which happens to occur in bacterial flagellas, and the 22 amino acid epitope of this flagellin is recognized by plant cognate receptor, FLS2, as you can see here, and that alone is it can induce plant immune responses. So with this controlled stimulus, we are applying it to one plant. So as you can see in this diagram here, this is depicting the two plants growing side by side in these mesh pockets, and we stimulate one of the plants, which we call sender plant, with wounding and flagellin, or treat with water for a control. And then we look how the receiver plant responds. And very interestingly, what we found is that when you put Botrytis cinerea, a necrotrophic fungal pathogen, also that Javier showed you in the earlier talk, the receiver plant is becoming more susceptible to this plant when it's receiving signals through this fungal network from its neighbor. And curiously enough, when you use a different plant pathogen, in this case we used Fusarium sporotrichoides, you see an opposite trend. You see an induced resistance in the neighbor. So there appears to be a species specificity. And this again, if you were here with the moment ago, in the talk, Javier explained this also. He observed that with the trichodermis, and this I think is very fascinating and emerging topic in plant pathogen interactions, that there might be very specific species-specific effects through systemic signaling by trichodermis, or in this case, as I'm showing you, through these mycorrhizal networks and neighbor signals. And as a control, we also interrupted the connection between the two plants by cutting the mycorrhizal network. And when we do so, we actually abolish this susceptibility or resistance phenotype. And dare I say, as a control, we also did a control cut in the first treatment here, but that was not done between the plants, but on the side of the plants. And with this, we wish to control for the effect of touching the fungal mycelium itself. So what is different between A and B is really the positioning, where we touched, whether we interrupted or didn't interrupt the connection between the two plants below ground. What we are interested in the lab to do is really understand what are the molecular responses of these receivers, and that could explain these very interesting phenotypes. And in a same experimental setup, we harvested the leaves from the receiver plants to look at the gene expression in the connected and in the cut systems. There are a lot of genes changing up and down in this, but what we are interested in is really seeing what is the difference and what is the connected treatment unique genetic signature. And so here, these are the differential expressed genes in the connected and cut treatment after the stimulation with wounding and flagellin versus the control, water control. And we can see 4,800 or so differentially expressed transcripts being specific to the connected part, and thus potentially explaining the phenotypes of the fungal susceptibility and resistance I demonstrated earlier. And what we do, we want to identify what are the functions of these gene sets. And this we do a lot of function, also de novo functional kind of annotation and grouping. And we here we compare those functions that are unique to the connected plants, which you see in the first two columns, compared to the cut plants, which you see in the other two columns. And what is strikingly emerging that first, there is a difference in the photosynthesis, genes regulating photosynthesis. In genes that fall into RNA processing and protein biosynthesis. And then another interesting cluster, which is enriched with transcripts that are involved in plant defenses and also different enzymatic reactions that might be directly or indirectly related to that. And when we look in detail, and this is manually annotated plant defense kind of landscape where all the known components of pattern triggered immunity, effector triggered immunity, MAP kinases, and the signaling cascades, as well as many transcription factor families known to be involved in plant-insect interactions, are depicted. We also depict things like genes and enzymes that are responsible for ROS generation and homeostasis, or the ubiquitin ligases and proteasome activity. And we can see that there is quite a considerable remodulation of some of these components in the gene sets that are specific to the connected plants and receivers. We also see remodulation of plant cell walls, and specifically also jasmonic acid and few other plant hormone responses. On top of that, we also expanded this analysis and looked at the compounds that are changing in the receiver leaves. And interestingly, so also you can see in these volcano plots compounds that are going that are overproduced or underproduced in response to wounding and flagellin, that you can see above the line in the volcano plots here on the right or left. And interestingly, when we compare these sets of compounds between the cut and connector treatment, we can see that these are different compounds. And hence we decided to look at the relative production of the compounds from our HPLC-MS data and volatile data that we could identify. And we wanted to see which are the relative strongest differences between the connected and cut treatments. And what emerged actually is a sort of a cluster 1 and cluster 2 of compounds that show opposite trends. In one case, which are produced more or maybe suppressed in the cut and produced more in the connected, and vice versa in the cluster 2. And the type of compounds that you can find here, they involve monoterpenes and triterpene compounds, such as saponines, which have described roles in plant pathogen interactions. And then we also wanted to ask a question, how did metabolic and transcriptomic data correlate with each other? And can we find metabolites that have the strongest correlation with certain genes in our connected plants, and what those genes are? And this actually, by doing so, we actually identified these 4 compounds that show the strong correlation with quite a limited subset of transcripts, which I depict here in this network. So here you can see 2 compounds in particular, proline, which has known roles also in plant ROS modulation, and one of the triterpenoids, soy sapogenol, often found in leguminous plants. And the lines drawn here indicate positive or negative correlation above the threshold we set with transcripts that show differential regulation in the connected plants. And quite curiously, here we see quite a lot of actually genes that are involved in plant pathogen interactions, such as the ubiquitin, E3 ligases, pathogenesis-related proteins. We even see the FLS2 receptor. And then we see also quite a lot of transcription factors from MIB and GRAS family, which could have potential roles in the metabolism and then be part of the plant defense pathway. Without going too much into detail in this, I want to illustrate that we are really touching the surface here and opening up a brand new field of research, and definitely for us. And in this image, we depicted kind of the landscape of different interplant signals and components involved in this. What I showed you here before is kind of the receiver responses in receiver leaves. But what we do not know yet, and what is the subject of future and ongoing investigations, is what actually are these signals? What are those mobile signals that come from the sender plant, go into their roots, that go into the fungal network, whether fungus is responding to these signals, modulating them, or creating additional signals that then are perceived by receiver roots and then systemically inducing responses in above ground. So our the vision of my emerging group is really to try to approach each of these components in a stepwise fashion and try to identify these things. One of the ongoing projects that we have is looking at the hypothesis that small RNAs could be potentially involved in this interplant signaling, and we are sequencing the small RNAs generated by the sender plants and comparing those to the receiver responses. And I really hope that in year or 2 years' time you will see more about that. We have a very strong collaboration with the Latvian State Forestry Institute, and I list some of my colleagues here, and we're actually utilizing a very interesting resource that I'm depicting here if anybody is interested to reach out and collaborate on that as well. These are hybrid aspen and silver birch, and we can grow them in vitro cultures in sterile, clonally propagated plants, really to use selected clones for specific experiments. And because these two plants have genome sequences available, we are actually doing our first preliminary experiment of, for instance, characterizing the immunity-related genes and their activation in systemic leaves of these plants after eliciting responses in another leaf. This is just to illustrate how we do this, and that we can identify some of the immunity components and find their homologs in each of these tree plants. We also are very interested to explore the microbial diversity of mycorrhizas, and we, for instance, have looked at the hypothesis that the silver birch and hybrid aspen productivity could maybe be explained by differential mycorrhizal communities. And contrary to our hypothesis, we actually found that there is a more difference in diversity in silver birch that doesn't show any difference in yield performance versus hybrid aspen that showed clear differences in yield but didn't show any differences with the mycorrhizal community composition. And so this is another aspect in the lab that we are trying to do. We're really trying to develop this these tree models and expand on the work on trees, looking at the microbial diversity in tree roots, and then doing functional experiments in the lab similar to those that I showed you with Medicago. And trees are really important crops for the Nordic economies, and hence that's a great motivation for us to do it. And the final aspect I wish to illustrate here is a new project funded by European Regional Development Fund in my group, and in collaboration with a company and Agriculture Research Institute in Latvia, is our ambition really to explore the diversity of soil microbes. And by this, we want to start by taking soil samples and trying to isolate novel mycorrhizal fungi that are endemic and specific for our region and indeed the Nordic region, looking at their diversity, describing the communities through ITS sequencing, similar to what I showed you in trees, and really try to cultivate in the lab. And for this, we have the carrot hairy root cultures, which serve as a lab host for new mycorrhizas. And so this is again a kind donation from our partners and Sunjay Lee in Switzerland, and really thanks for this has enabled us to do a lot of work that I've been showing you before here. With the aim to test these novel isolates in crops. So the conceptual idea is here that we could actually try to harness these common mycelial networks for crop protection. So if you imagine that there might be native endemic connections, and when one plant is being attacked, it might send the signals to the neighbor plants, and our experiments do support such a phenomenon. And if we understand what these mechanisms really are, we can perhaps engineer a better signal senders that we can plant as a sender plants for these other crops. And if we have locally adapted other mycorrhizal products, they could really make stronger supplemented connections between these plants to really achieve community level resistance. When these plants are attacked, then they might have kind of heightened defense capabilities. And here we also, of course, have to explore when these these responses are actually heightening or weakening these responses, and how crop specific they are. So all in all, our lab is really starting off now by addressing these fundamental questions using model system. What are the molecular mechanisms in inter-plant signals, and really trying to aim for more applied applications in long term, trying to engineer the signal decoding or production to enhance resistance in important crops, including trees, and also employ novel microbial preparations. And we have also ideas with plant growth promoting bacteria as well, similar to mycorrhizal fungi. So we're not only limited to in our ideas to mycorrhizal fungi. And with that, I want to finish with a very light but very important aspect that we are very passionate about. We are very passionate about public engagement and communicating science, plant microbe interactions, root microbe interactions to community. And we do this by scientific art. This is one way which has been a passion of mine, which started in Switzerland, actually, where we produce artworks like you see here, which depict different soil microbes or plant interactions with insects. And these artworks are displayed in art galleries. We take active part in the European Researchers' Night and all other events where people from my group are really telling general public, school children, everybody who is interested in about these interactions, and do a few demonstrations as well. So with that, I would like to take huge thanks for the organizers to invite me and introduce to our new group. And if you're interested, you can please follow the website, and I'll be happy to take any questions that you might have. Thank you very much.
Thank you very much for the very nice presentation, Zigmunds. It was lovely, a lot of very amazing pictures. So it was very nice to follow. I already see a question popping up in the chat from Victor. A question on how you can exclude the impact of microbes rearrangements and also the soluble and volatile compounds released to the soil.
Yeah, an excellent question, Victor. So actually, this is one of the rationales that we had in the experimental design, that we do not wish to specifically create an artificial system that lacks volatile components and that lacks soil exudates. In nature, all of these signals, in our opinion, might coincide, right? It's very well established that volatile compounds can mediate plant systemic responses within the plant as well as between the plants. And what we are looking is, what is the contribution of the mycorrhizal networks and their connectivity on top of these other types of wireless connections that happen there. So we do allow for the root exudates to happen and the volatiles to happen, the exchange to happen. And as you saw from my experiments, we really dissect out the contribution of the connectivity of the mycorrhizal network by either allowing it to be connect to plants or cutting it in half. And that alone, in the background of all other signals, demonstrates a very interesting difference, which we then want to pursue and identify what are the components and what the functions of them are.
Okay, that's very nice. I think that answers the question. I see also another question popping up by Carmen in the chats. So how do you reconcile mycorrhizal networks with annual crops replanted every year, unlike forest, and in highly disturbed agricultural environments?
Yes, of course, that we know that mycorrhizal fungi and establishing networks between plants requires some time. In our experiments, that happens in 8 weeks. So it is a relatively short time, and if we consider in field during one growth season, it is reasonable that this can happen. So what we do not know how really far these networks extend in agricultural systems or in nature, or how fast a fungus can grow from one plant to another plant. This is something actually we want to test in the lab and maybe model how it could happen in the field. But I think AMFs and AMF products are applicable, and they earn the interest to research them also in annual crop systems. And in trees, we specifically focus on broadleaf trees. Aspen has been reported in the literature to form endomycorrhiza associations on top of the ectomycorrhizal associations, which historically have been associated with trees. And due to the climate change in the northern region, more and more broadleaf trees will be planted and be growing here. And also there is this succession from ecto to endomycorrhizas. So what we believe is really an anticipation of the climate change, that these changes in communities will become more and more relevant and important.
Thank you. I also see there's a lot of questions popping up in the chat. So yeah, I talk with a lot of enthusiasm. So I also have a question from Olga. Do you think mycorrhizae are actively involved in signal transfer? And if yes, what is the evolutionary purpose of them being involved in this transfer?
I can maybe answer brief to Olga. These are exactly the same questions that we have in the lab, and we really hope to explore them in the future. Yes, we do think that mycorrhiza could be picking up the signals from the sender plant, maybe modifying those if they are metabolites, or if that changes the fungal gene expression, then the fungus could be creating kind of secondary signals that propagate. This we want to find out, and also with the small RNA experiment, we want to map the small RNAs that we find back onto the fungal genome, which has been sequenced to identify potentially fungal origin small RNAs. So that's one avenue. About the benefits, this is a very interesting question, and probably goes way beyond the minute answer. But I think there could, if you have a better protected or nourished host, then the fungus might be benefiting as well from associating with such a host. Also, if the signals from one plant help the other plant, and if the first plant is being attacked, then the fungus can kind of maybe even shift the resources and affiliate more with the sort of the neighbor, more protected host. So there could be a lot of beneficial interactions, but also we have to, consider all other scenarios with simply neutral and also negative effects for both fungus or these plants, as in the case with susceptibility. If these signals elicit susceptibility, that this could be a mode of sort of kin competition rather than kin selection and benefits.
I think that definitely makes sense, that you just want to be surrounded by as many good hosts as possible to a certain extent. I see that no more questions are popping up, but I do see Rafa appearing. So do you have any questions?
I have one question. So I don't know if you say before, but how long do you need to wait to see the response in the receiver plant after you induce the wound?
In the Medicago system,
how long it lasts?
All right. So what I can tell you is how when we harvest the receiver plant. So these are 3 days. So 3 days after stimulating the sender, we harvest the receiver, and then we see these responses. How long they last, we really don't know. That would be an excellent future question.
Okay. Thank you.
I have a question, if I may.
Yes.
Thank you, Zigmunds. It was a really, really nice talk. Really excellent work. Congratulations. I have a basic question concerning the sender and the receiver plants. Are exactly the same variety? Maybe I missed it.
Yes, they are exactly the same ecotype, genotype of Medicago truncatula R108. So we are looking at intra-specific interactions and then extending that, whether similar things happen between species. That again, I think, is an emerging question in the field and will be answered by several papers maybe in the next couple of years.
Yeah, I was wondering about this. I mean, in my line of research, it's more the allelopathic interactions, right? And then we see that it's not the same to have one wheat variety with another wheat variety, or the combinations. And of course, this is really much more complicating and expanding tremendously the amount of work. But maybe in future research, you can have a look.
Yeah.
Thank you.
Okay, thank you everyone for again a very nice discussion, I think. We of course had very nice talks as well that just got a lot of questions surrounding them. So thank you all very much for joining. So I think that is it for now. I don't see any more questions popping up in the chats. Okay, so I think that was it for us today. So for the next talk, we already know that Professor Luisa Lanfranco will be giving a presentation, and the other one is still to be announced. Yes, to be announced indeed, because we had to switch a little bit in our scheme for the last two talks, with Zigmunds nicely accepting our invitation to talk already today. But yeah, we will make sure to send you our updated schedule as soon as we can. So thank you, everyone. And thanks to our speakers again as well.
Thank you.
Thank you, everyone.
Bye.
See you at the next one.
Thank you, everyone, and have a really interesting talk. I really enjoyed it. Naomi, thanks, and Raphael, and see you in other sessions. Thank you very much.
There you go.
Sure.
Thank you very much for the talks. They were amazing. Yes.
It was really great.
Thank you.
Okay.